My name is Stephen Chopski, and I'm a chemical engineer in the Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs at FDA. It is my honor and pleasure to introduce the second session on day 2 of the Model Master File Workshop, MM applications for locally acting drug products. We'd like to ask you to reserve your questions for the speakers until the panel discussion. We'll have a panel discussion following the lunch break. Session 2 will offer case studies and engage in discussions about situations in which an MMF can support product development and regulatory submissions for locally acting drug products, including orally inhaled drug products applied on the skin, and ophthalmic drug products. Our first speaker is Dr. Ross Walenga, who is a chemical engineer in DKMM ORS at FDA. His research interests include computational fluid dynamics modeling of orally inhaled nasal, ophthalmic, and dermal drug products to answer questions pertaining to bioequivalence. Our second speaker is Dr. Flora Musuamba, who is an associate professor of clinical pharmacology at the University of Namur and the University of Musuamba. She is a pharmacometrics and pharmacovigilance internal expert at the Belgian Federal Medicines Agency and a member of the EMA. Our third speaker, Dr. Jan de Bakker, is the founder and CEO of Fluida. He is an innovative researcher in the field of airway modeling and in the field of respiratory and sleep medicine. Our fourth speaker for this session will be Dr. Markus Fridén. He is a senior principal scientist in the Department of Inhalation Product Development at AstraZeneca. He's a specialty in mechanistic modeling and simulation of inhaled drug products. Our fifth speaker for this session will be Dr. Abdullah Hamadeh. Is a research associate in the School of Pharmacy at the University of Waterloo. His current work focuses on the development of open source computational tools for robust in vitro and vivo extrapolation and virtual bioequivalence assessment. Our sixth and final speaker for this session will be delivered by Dr. James Clarke, who is an associate principal scientist in the SimSet division of Sujatha. He has worked to develop and enhance dermal absorption in PBK models. So the 6 talks will be followed by a panel discussion where our speakers will be joined by Dr. Jessica Spires, who is a principal scientist at Simulation Plus, Dr. Mingliang Tan, who is a senior pharmacologist in DKMM ORS at FDA, and then our final additional panelist will be Dr. James Mowli, who is the Director of Scientific Affairs at Capstone Development Services. The panel discussion following the lunch break will be moderated by Drs. Khondoker Alam, who is a senior pharmacologist in DKMM ORS at FDA, and Dr. Sujatha, who is the Vice President of the Drug Product Development at GSK. With that, I would like to welcome Dr. Ross Walenga to the podium.
Okay, thank you. All right, it's my privilege here to be speaking to you today about regulatory perspective on MMF applications for OIDPs, ophthalmic drug products, and drug products applied on the skin. The little disclaimer, this reflects my views and should not be construed to represent FDA's views or policies. So I thought before beginning the session on locally acting drug products, I'd start with a brief definition. So these are drug products not intended for systemic delivery, although they may make it into the systemic circulation. And they include a variety of routes of administration. These include buccal, gastrointestinal, intrauterine, nasal, orally inhaled, ophthalmic, otic, rectal, applied to the skin, and vaginal. And of course, the focus of this presentation will be orally inhaled, ophthalmic, and applied to the skin. So starting with orally inhaled drug products, or OIDPs. These are products that are typically indicated for diseases such as asthma, COPD, cystic fibrosis, even influenza, and others. They also include systemically targeted products, but that's not necessarily the focus of this presentation. And the device types, like you can see in the figure here, include meter dose inhalers, or MDIs, dry powder inhalers, or DPIs, and inhalation sprays, which would be aqueous droplets that are actuated with a spring-loaded mechanism. So why might you build a model for OADPs? You might build a regional deposition model, such as using the computational fluid dynamics, or CFD, or a semi-empirical method to predict the impact of device or formulation changes prior to your in vivo or in vitro testing. And this could then accelerate product development by avoiding unnecessary testing. And then for generic OIDPs, modeling could be used to justify biorelevant bioequivalence limits, or BE limits, for relevant in vitro studies. Another potential use would be with physiologically based pharmacokinetic, or PBPK models. And this could be used to examine the relationships between in vivo plasma concentration, PK data, and regional absorption. And then this could be used to make decisions about product development based on PK data. So I'd like to discuss a couple of potential applications for MMFs in this space. So one idea would be to develop a regional deposition, CFD, or semi-empirical model for any given device type. So you would focus on either MDI, DPI, inhalation spray, and then reuse it for other drug products in that same kind of class without the need for extensive validation, model credibility. Another potential application is intravenous or IV or an oral PBPK model for a given active ingredient. And this could be reused for the development of an OIDP PBPK model for other drug products that include the same active ingredient. So diving a little bit more into these examples, this one will be a CFD model for MDIs. So this image here is from an internal project that I've been working with. And the model purpose could be to develop biorelevant BE limits for realistic APSD testing. And the physical models could include thermal energy, species transport, turbulence, et cetera. And so this could be a really important part of the MMF because for a physics-based model, a key part is defining which physics models you're going to include. So this could be the part that kind of stays the same from one product to another. Your model inputs could include realistic APSD and plume geometry data, so that might not be part of the MMF because that might change from one product to another. The mesh and time step sensitivity would be established for a physics-based model. Establishing your discretization is an important part and takes up a lot of time, so this could really save time by including this in an MMF. And your model would be validated with in vivo nuclear imaging data for multiple drug products. So again, the data source could be part of the MMF. So going a bit further with this application, this is another image from this project. The selection of the single human airway geometry or a set of human airway geometries could be part of your MMF because you may be using one airway model or multiple airway models. And for the verification and validation, since this would already be established, if you've got a similar enough product, you may only need to do software quality assurance, which is just basically running the model scripts and making sure that it's working properly. If you were to use it for another drug product, if it's in the same class, I think that they would be easily used if you were going to, say, solution-based MDI to suspension-based MDI, or from, say, single active ingredient to multiple active ingredients, you might need some kind of bridging to then use your MMF for this other application. The other one I mentioned was an IV PBPK model. So this was from another internal project for budesonide. So this could be applicable to any PBPK model that includes the active ingredient, so not even necessarily OIDPs. If you've got budesonide in another product, and I know budesonide is used for other dosage forms and routes of administration besides OADPs, it could be useful for that too. And then this would include the IV PK data or the sources where the IV PK data were taken from. Then the output would be the clearance and distribution parameters, and this is what you would use for your MMF. Next case here is ophthalmic drug products. So these are administered through the eye, typically either through a topical application or through injections. They're indicated for a variety of eye diseases, and typically you're trying to target certain eye tissues or eye regions. These encompass a wide variety of dosage forms, including solutions, suspensions, ointments, etc. So why might you build a model for an ophthalmic drug product? So like any other PBPK model, you can mechanistically describe the release from the active ingredient to the different relevant tissues. You can then correlate in vitro metrics to in vivo exposure. There could be a lot of reasons for doing something like that. You could then assess BE between the RLD and the test product. so a virtual BE simulation. Or you could use preclinical data to inform product development by doing interspecies model extrapolation. So for ophthalmic drug products, pretty obviously, it's hard to get, or really impossible to get drug exposure at the site of action, ethically, of course. But there have been a lot of rabbit models developed for ophthalmic drug products. And so by using these rabbit models, you can then maybe do interspecies extrapolation. So some potential MMF applications for ophthalmic drug products. So one could be to reuse the validated model components, especially for the drug diffusion and partition into the different ocular tissues, and then maybe going from one dosage form to another. The other potential use would be to establish the anatomical and physiological parameters that you're using for your model, and then you can use this across many different ophthalmic drug products. So kind of similar to a platform validation. So this first example, the same diffusion and partition parameters across dosage forms. So the purpose could be to predict changes in ocular bioavailability and PK due to differences in the formulation, such as particle size distribution, viscosity. And this example here was an ocular model developed for dexamethasone suspension. And this is a published result. You can see the validation in the different ocular regions, cornea, conjunctiva, et cetera. And then you can then take the same diffusion and partition drug parameters and then apply them to, say, an ointment model. So in this case, this was actually taken to an ointment model, and you can see the validation here. So they took the same parameters and then moved it to the different dosage form. The overall ocular model would be the other example. So the model purpose could be to demonstrate the predictive ability of this organ model. And then the anatomical and physiological parameters, such as tissue volumes, surface areas, tear film and tissue thicknesses, that could be what's in your MMF. So you can see an image on the right. This is for thicknesses in the cornea. So if you look in literature, you might actually find a lot of different values for what these thicknesses would be, but your MMF would then maybe establish what this should be for your model going forward. Then you could have platform validation with multiple drug products across multiple dosage forms to establish the predictability of this. And then the end goal is that you've got these parameters that you don't need to keep kind of rethinking. So lastly, I'll speak about drug products applied to the skin. So these are applied to the outer surface of the skin, and they can target the local tissue or systemic delivery, which is not necessarily the focus of this presentation, but will still come up a few times. So these encompass a wide variety of dosage forms, as with ophthalmic drug products. And the topical and transdermal delivery systems are indicated for either local delivery or systemic delivery. So you can see an image of what these things look like. You have ointments, paste, powder, oil, et cetera. So many different dosage forms. So why might I build a model for modeling for products applied on the skin? Dermal PBPK models can mechanistically describe the active ingredient release and for delivery to the skin under in vivo and ex vivo scenarios. And you can then use this to identify a safe space for in vitro metrics related to the quality of the RLD product, and then assess the impact of these deviations. You can predict skin permeation in a IVPT kind of in silico model. And you can assess BE between the RLD product and its prospective generic locally in lieu of conducting an in vivo BE study, especially when this might be challenging to conduct and could potentially decrease unnecessary human testing. So a couple of potential applications for products applied on the skin. First would be to develop a systemic disposition model. So this is similar to what I talked about for the OIDPs when you have the IV PK model. You could have validation of a computational framework for skin absorption in virtual subjects or for an IVPT kind of model. Or you could have an IV-IVE methodology for taking this IVPT study data and then using it to determine parameters related to skin permeation and drug product in vitro metrics for your in vivo model. So the first example is validation of a computational framework that I'll discuss. So this is to demonstrate the predictive ability of this framework. The credible computational framework is then going to increase confidence. And then some things to think about when building this case example. So the first thing was that the dermal PBPK models were applied under many different dosage forms. And the active ingredients were selected to cover a wide variety of physicochemical properties and PK characteristics. And the selected skin products were consistent with the complexity observed in the drug product of interest. So this was a published case. This was diclofenac sodium. I think many of you are familiar with this case. And the applicant supported their modeling and simulation approach with a validated drug product specific dermal PBPK model and the validated framework. And to establish the framework, they looked at many different dosage forms, including TDS, solution, cream, gel, ointment, et cetera. Also looked at many different verification matrices, including plasma, IVPT, synovial fluid, and others. So once you've got this validated framework, you can then apply it for topical cream, topical lotion, topical gel, or TDS. So final example here would be IV-IVE methodology. So to develop an IV-IVE methodology that leverages IVPT study data towards the development of an in vivo dermal PBPK model. And some things to think about when building this case example. The IVPT study data are available for certain products, and they reflect the impact of drug product quality attributes under ex vivo conditions. You would then leverage IVPT study data to establish the IV-IVE and inform permeation-related parameters or other uncertain parameters. And then you're using this to develop your in vivo PBPK model. So appropriate model validation of the in vivo model is still necessary before being applied for population predictions. So this case was presented last year at ASCPT 2023. And it was describing a workflow for a ketoconazole model. So you take an oral PBPK model for ketoconazole, a skin absorption model for ketoconazole, combine them, and this creates an in vivo dermal PBPK model. You then take your IVPT study data to inform your influential model parameters, and then you can describe skin permeation, and then you use this optimized model to develop a safe space, in this case for viscosity, so you can show the impact of differences of viscosity on PK. So I've got a few conclusions from today's talk. The mechanistic models have been used to support product development and approval for locally acting drug products such as OADPs, ophthalmic drug products, and drug products applied on the skin. They may be generally useful for understanding relationships between in vitro metrics and in vivo exposure, or for conducting BE trials, virtual BE trials. And I presented several examples that propose the use of MMF for these classes of locally acting drug products. And so I'd like to conclude with acknowledgements from many people across our office who have helped with this, but especially want to call out Mingliang Tan and Eleftheriot Sakalozu, who both contributed substantially to the slides that you see here. They really are our experts in ophthalmic drug products and products applied to the skin. And I've included some references for your reading later on.
Thank you for your excellent talk, Dr. Walenga. At this point, I would like to welcome Dr. Flora Musuamba to the stage here to present her presentation.
Yes, I would first like to thank the organization. This is a great meeting, and I'm having great time here within the meeting and around the meeting. So thanks for the kind invitation. So I have a disclaimer here because, you know, I'm working at the Belgian Medicine Agency, involved in activities of the European Medicine Agency, but what I would be discussing here is only to put on myself. This is the outline of the presentation. I would start by some introductory word on the qualification procedure that we do have within the European Medicine Agency. And then I will share with you our experience, where are we, and how I see that connecting, so to speak, with what we have been hearing since yesterday about the MMF and even the fit for purpose that I was presenting previously today. So what is it about? They asked me to share our experience with what we call qualification of new methodology. This is a sort of procedure that have been put in place by the European Medicine Agency to support the drug development program, and in particular, to set up a sort of context for sponsors or CROs or consortia to come and discuss with regulators in a sort of non-competitive setting, outside any kind of pressure due to a certain drug development program, timeline, or whatsoever. The aim of this kind of procedure is really discussing amongst methodologies. The particularity of this procedure, when you compare it to the other procedure within the European Medicine Agency, this is where we are allowed as regulator to invite the expert, the key opinion leaders from outside, from academia, to come and support us. When someone come and put on the table a biophysics model or a PBBM model, that's not that different for me, actually. And I do not have that particular experience. For that one, EMA will find me, go around, and find out someone who is strong in biophysics and reinforce my team. And in the qualification team, you have, in average, half-half regulators and external experts. Really, the aim is discussing at methodology level rather than in a particular drug development program level. Who can apply? Most of the time, I'll show you in my upcoming slide, these are consortia. So a publicly funded group that would come. We have also some applicants being pharmaceutical companies, couple of them. We have also had the MCP mode that are presented. We have software developers. We have had SimSip coming recently. So it can be whoever. If you think that you have an innovative methodology, and most of what we have heard since yesterday would qualify, so to speak, for that. Yesterday you heard me inviting Professor Uker to come and submit what he was presenting here for bioequivalence, etc. So this is really something which is offered at methodology level rather than at a particular drug development level. What is also interesting to discuss here on the right-hand side of the slide is the fact that for the applicant that make it, from our side, there is no debate. It goes public. If you make, and it goes public not on the CPAS somewhere hidden, no. It goes public on the EMA website. We publish the model, the code, everything. And we publish that actually twice. First, when we have discussed with the applicant and we kind of reach a sort of agreement from our side. When I say our side, I mean the qualification team side. We think that this platform qualifies for us, for the context of use that was discussed. Before we give the final stamp, it first goes public. And then we invite other people that think that they're experts in the field and were not part of the qualification team to challenge the model. Comment. This is where we're going. Do you think that this makes sense? 3 months public, and then generally we receive comments, we update, we discuss with the applicant, and then we release the final qualification opinion that also goes public, along with the response to the comment that we have received. So this is the way it works. But like someone was discussing yesterday, not all of them make it to the opinion. And we have a sort of midway part which we call the support letter. It happens that some consortia, some applicants come and request for an opinion. We see value from what they're proposing, but we do have problem with the validation data set. We think that some of the components of the model are not mature enough. We release what we call a letter of support that also goes public. We say we like the idea of using a model-based approach for this application. But from what we have seen, we think that this needs to mature on this or that component. This is actually the way it works. So we also can release a letter of support. The opinion is public, the letter of support is public, but the data, the code, the model behind the letter of support is confidential. Unfortunately, I just need to share with you that some of the sponsors play with that. They come and request only the qualification advice because they don't want to go public. They just want to have the support of EMA. And then they never come back. This shouldn't happen, to my view, but actually this is the way it is. In terms of the different regulatory procedures that we do have, we have what we call the Innovation Task Force briefing meetings. This is something which is offered primarily to academia, SMEs, that have less of experience with regulatory setting. These are informal meetings to sort of prepare the submission for the qualification. And then we do have scientific advices. I will show you an example. Normally, the scientific advices are dedicated for a particular drug development program. The sponsor have the right to say, for this drug development program, I have qualified this particular method, modeling and simulation method, and this will be included only in this drug development program. We can also discuss some methodological aspects within the context of a scientific advice. We do have these qualification advices and opinion procedure that I have already discussed in my previous slide. And then we do have the marketing authorization application. There also, it happens that one wouldn't have a qualified method, and they come with that for the first time. This is not my preferred, but they can do. At the time we're discussing the approval of the medicinal product, we're also discussing some methodological aspects that support dose selection or bridging across formulation, things like that. Everything at the same time. It's not ideal, but frankly, not firmly forbidden. Just to say that this qualification procedure that I'm presenting here, for modeling and simulation tools in particular, it is actually mentioned in some of the ICH guidelines. This is an example, ICH guideline for assessment of QT liability. It is really mentioned that some of the regulators do have a qualification procedure in place. Please, if you think that you have an innovative method that can, replace concentration QT analysis or ERG assay or whatsoever in vivo telemetry studies, you think you can bypass that with a modeling and simulation method, go for qualification. It is really explicitly mentioned in there. We have some guidelines at EMA that also recommend qualification. The PBPK guideline is one of them. And there we are clearly saying that for high impact, I told you about high risk. I will be using the impact just to make it a little bit different from FDA. But most of the time, the high impact are also high risk situation. But it happens that the impact and risk are slightly different. I'm coming back to that. We recommend for high impact application of PBPK to qualify the platform being the system model. And I like a lot the discussion I heard yesterday, distinguishing the system model, the drug model, and the formulation model. And what we're insisting on here is really the system component of it. Please come with that. It has also happened that some of the applicants, they came for advice during the drug development program before the confirmatory studies. And then during that scientific advice procedure, they are asking a question that is more on the qualification setting. One of the examples is on the screen. They are developing a drug which has a very high protein binding level. And then they are coming with a PBPK model. But they turn their question subtly. They say, can the agency agree that the PBPK approach that we're proposing here would be sufficient to adequately assess the DDI liability for highly bounded therapeutics. So they go outside, actually, their compound. I suspect that they have a couple of 2, 3 other therapeutics out there, and then they want to have the yes already here. We say yes, we see some value, in particular for the drug that is on the table now. But if you want to engage the discussion beyond this drug development program, come for qualification. Then we discuss openly for all the other drugs as well, because in the context of the scientific advice, we need to stick on the drug development which is concerned. And this has a lot of commonality with what we have heard so far. We are pushing for qualification of modeling and simulation approaches, particularly for reusable model. What do I mean? I mean the model that are intended to be used for more than one drug development program. This is where I see the value of discussing this methodology. We also see the value of that for complex model, and we also see, of course, the value using that for what we call high regulatory impact. High regulatory impact is an application where you have these modeling and simulation approaches, which is proposed sort of to replace something which is established as an evidentiary standard for that kind of question, that kind of context of use. To establish pivotally efficacy, safety, one would expect to see a randomized controlled trial. Now we don't have that. We have that in silico virtual digital twin. I'm not saying can't make it, but I'm saying this is an high impact. One would expect to see a placebo arm. Now we have a virtual arm. One would expect to see a bioequivalence study. And now we have this modeling and simulation approach, and we think that this kind of should drive a little bit of the scrutiny and the seriousness we put into that as regulator. Now let me share with you some of the experience that we do have. We have seen a lot of qualification and requests, and this is increasing. So this is kind of stopping the statistic last year, and you see that it is increasing with time. Most of the time, we do have SMEs coming, but we also have consortia and pharma coming to that. The scope is quite wide, going from preclinical development tools, and now you know, because we are very supportive of the 3 R's, we have now some couple of qualification procedure ongoing where some of the non-clinical animal studies are proposed to be replaced by some sort of in vitro supported modeling and simulation approaches. We do also have a lot of qualification coming for dose finding, enrichment of population, validation of endpoint, etc. And these are kind of scope that we do have. Sometimes they make it to the opinion. Most of the time they have at least the letter of support, and it happens that they have nothing. I will show you one example in a few minutes. When they file for a qualification opinion, in general, they do have questions. And the question that we need to agree on for all the opinions are related to the context of use. The context of use needs to be very clear. We need to have a very clear view on the space where the applicant is planning to use the qualification method. We cannot give the stamp. I mean, this is a little bit the difference I see with the fit of purpose, where the door is kept open. Like you can go and improve it, and when it improves, it can do even more than what we have seen now. We want to be a little bit more clear on where are you planning to go with that model. And only what we have agreed on as a context of use would be applicable. We discuss on the data. The context of use needs to be supported by the data, but most of the discussion is going on the methodological part of it. We discuss the link between the question of interest, the data proposed, and the context of use and the methodology. Yes, most of the time we are in disagreement with the initial proposal related to the context of use. We discussed that. It matures with the different interaction we have with them, the data, the methodology, etc. It's a kind of progressing exchange with the regulators. So I will only discuss two examples, one that didn't make it and the second that made it. The one that did make it, a consortia, public money, they come with a qualification advisory file for dose selection in the field of osteoporosis. It was a quite strange application because they were proposing to qualify a model that they were not intended to use for drug development. It was like a sort of academic exercise just to press the regulatory system and to see how it works. And when we told them that we did have a problem with the validation data set, in order for that model to be acceptable in the regulatory setting, we would like to see more of the data for clinical validation. They responded that we don't see anyone putting money into generating data to validate a model for those selections. They did come with the model for those selections. And then they say that they don't see the value of investing for something also only serving for, it was a little bit strange. And then we said that because we are here to help drug development, your model will not help drug development eventually. Maybe it's even not worth go into technical discussion, etc. So that was really a very negative one of the most negative I have seen in my 10 years working at the agency. A positive one is the one that we gave a positive opinion to CPAS Institute. They came with a model-informed approach to support enrichment biomarker for type 1 diabetes. It was really a model that matured during the interaction that we did have with them. In the end, they updated their context of use, which we agreed. They also brought more of data than what they were proposing in the beginning. We discussed that. We worked on the model together, and in the end, we agreed on the data. We agreed on the covariate that were proposed, and the different, the model was qualified and is now publicly available on the EMA website. If you go there, you'll see all the codes. Everything is there. Even the analytical method that they're used for measuring the biomarker, it is all described the way we received that. So someone who wants to replicate that model should have enough to do so. My take-home message, the take-home message that I would propose you for this qualification opinion is that it makes sense only for models that are to be used in more than one drug development program. Given the resources and time that it takes both sides, maybe let us put it only for the more complex one. Someone was saying IV one compartment, maybe not. And when we have doubt on the data sources, when we have doubt on the quality of some of the parameter value, etc., it was having that discussed outside a particular drug development program and maybe put it forward for high risk, high impact applications. This is what I could share with you as our experience with something similar to what you're calling MMF. Thanks.
Thank you for the excellent talk, Dr. Musuamba. At this point, I'd like to welcome Dr. Jan de Backer to the podium.
Thank you. Thank you so much. It's great to be here. I really enjoyed the discussion so far and happy to contribute some of the strategies that we can see for model master file creation, specifically in the field of orally inhaled products. with a specific focus on digital twins and in silico trials, something I think is getting a lot of attention these days. As we've been discussing at length over the last 2 years, the model master file is still a novel concept, but it seems that certain elements are emerging as characteristic of some of these MMFs. One is that it should be scalable, it should be usable for various FDA submissions for multiple products. The context of use is quite important, and the validation associated with that context of use also is emerging as an important element. And it's generally confidential, which I think it makes sense from different angles. So the context of use that we'll be discussing in this talk mainly focuses on the use of these in silico models in combinations with in vitro MPK studies to obtain a biowaiver for the clinical endpoint study for the approval of orally inhaled generics. So in other words, can we use these in silico methods to make a digital equivalent of this clinical trial? And in order to do that, we need to identify the key drivers for regional exposure. And Ross mentioned the regional exposure element in his talk, which is quite important. So there's a special focus on regional deposition of these orally inhaled products. Probably most of you know the 3 main pillars for bioequivalence studies in the inhaled space. One is the device and formulation, where we typically use in vitro tests to see whether the test and the reference are within the acceptable limits. We have the systemic exposure, where the clinical pharmacokinetic studies are used to, again, test and the reference product. And then the local exposure, typically in the guidelines, there is a recommendation for a clinical endpoint study using FEV1 to understand if there's the same effect of the test and reference, with a lot of limitations, of course. FEV1 is a flawed endpoint, but these studies are very large and very expensive. That's why not a lot of companies go that route, because the return on investment for a study like that is not always that clear. So the question is, can we get better drugs faster to these patients, specifically in the US market, by approaching some of the in silico deposition study to really show that the local exposure of these inhaled drugs is similar between test and reference products? And there are a lot of evolutions happened over the last few years, I would say, in terms of the in silico methods and the digital twin. So can we really make a digital equivalent of a typical patient and have a digital equivalent of a clinical trial? But for these methods to be dependable, reliable, and accurate, we need to make sure that our digital approach actually captures the essential parameters of the physical process they are simulating. We know CFD very well. For many years, been used in automotive and aerospace, quite well validated. So we know it works. You can simulate those flows and the deposition, etc. But we also know it's very dependent on the accuracy of the input parameters. So we really need to identify the key parameters for the deposition of these orally inhaled products, including the device, the drug formulation, the inhalation profile, as well as the lung structure and the function. So all of these categories need to be adequately reflected in a digital element. So for the devices, we can use computer-aided geometries or reverse engineer the inhaler by also taking the CT scan of the device. The drug formulation is quite well characterized using cascade impactor testing, etc. For inhalation profiles, we can typically measure how patients use those inhalers in clinical practice. And then the important part of lung structure and function, that's where we use functional respiratory imaging. Functional respiratory imaging is something that we've been developing over the last 20 years. It starts from the conventional CT scans. And I think one of the major shortcomings in modern medicine is that the vast majority of these scans is still only visually assessed by a radiologist. So in my assessment, they probably extract about 5% of the information that is contained in those images, and it's not very reproducible. These scans end up somewhere and are never being used afterwards. So what FRI does, it converts the 2D CT slices into 3D reconstructions of the anatomical structures. So we can look at airways, blood vessels, lobe volumes, etc., visualize them, but also quantify them. And by taking an inspiratory and expiratory scan, we can make these static images come to life and look at functional parameters such as regional ventilation, area resistances, and also the deposition of inhaled particles. So without the need to radio label them, just using the CT scan and the computational fluid dynamics, you can see where those particles end up for individual patients. Validation, very important. So in terms of the total lung dose, we compared the functional respiratory imaging deposition with gamma scintigraphy for several products in several diseases. And in general, you see quite a good agreement simply because you're using so much patient-specific information for these simulations. Also, on a regional level, the body of evidence is increasing, where if you compare in the same patients the FRI-based deposition with the SPECT-CT, in this case, you see that also on a lobar level, there's a good agreement between those two methods. So you do have a lot of information. You have a lot of variables from the CT scans, from the inhalation profiles, from the devices. And one of the key questions, I would say, is how important are these individual variables? Are they all equally important, or certain parameters drive deposition more than others? So in order to kind of get some insights, what we did is we did a sensitivity analysis using Monte Carlo simulations, using thousands of simulations, where we wanted to see what are the influence of the different input parameters compared to others. So in order for a design process or a development process that we can put our resources, our attention to the points that matter the most. And the way to visualize it typically is using a tornado plot, where the bar indicates how influential the input parameter is in terms of changing the output. And in this case, the output is the deposition. So we did this for different type of inhalers. So for dry powders inhalers, for instance, here you see the intrathoracic deposition and the peripheral deposition. So the deposition that goes to the lung, as well as the fraction that goes to the deeper lung. And these are some of the parameters that we tested, and some of the parameters that are obviously very influential, such as the fine particle fraction, they pop up. But also you see things like airway volume, airway area, so patient-specific parameters, both for the intrathoracic deposition side as well as for the peripheral deposition. So if you dig a little deeper and you look at those images from those specific patients, you can clearly see that, for instance, in COPD and asthma, these lungs are very different than healthy volunteers. So you typically see hyperinflation. These lungs are larger than normal, and you also see the airways tend to be somewhat smaller due to the inflammation in those areas. It's even more pronounced when we look at cystic fibrosis or idiopathic pulmonary fibrosis, where cystic fibrosis is still characterized by hyperinflation, so larger lungs, a lot of bronchiectasis, a lot of inflammation in those airways. But IPF is very much a restrictive disease, so you see smaller lungs, but again, dilation of those airways. So it makes sense that these disease-specific characteristics influence the deposition of inhaled particles. For metered-dose inhalers, again, the fine particle fraction on top seems to be quite influential, and as expected, the peak flow. So we know that peak flows are very important to determine deposition, and that came out of this analysis, where if you look again a little deeper, you see that the flow patterns that are generated in these upper airways can be very different, whether you use MDIs, DPIs. And the peak flow very much drives things like local turbulence, enhanced deposition in those upper airways. And the upper airway size matters. If you have a narrow upper airway, you tend to have more impaction in the upper airway versus patients with large or more normal upper airways. So narrow upper airways associated with increases in BMI, sleep apnea, and all these things. For nebulizers, interesting. So nebulizers, as you probably know, it's a device where you use several inhalation cycles to really get the product into the lung. And so not surprisingly, things like ventilation, FRC lobe volume, which is kind of a measure for hyperinflation, pop up, as well as the mean flow, so the flow around tidal breathing. And again, if you look at those images, so in this case, on the left-hand side, you see an IPF patient, on the right-hand side, a matched healthy volunteer. And the colors of the lobes indicate the relative difference in ventilation compared to the matched healthy volunteer. So we know that IPF really starts in those lower lobes, and that's why they're colored red. So they're relatively underventilated, and those upper lobes are relatively overventilated. So they compensate for the disease. So you do see a shift in these ventilatory patterns depending on the disease. You also see something similar in COPD, where again, the upper versus lower ventilation changes. And of course, if ventilation changes, which is the main driver for deposition, we would also see differences in local deposition. So validation, of course, is a very important part. We need to be able to trust that these models are a true reflection of whatever it is that we're trying to simulate. So if we try to create a digital equivalent of a clinical trial, we should show that this indeed is the case. So I think there's quite strong evidence that the total lung dose can be predicted quite adequately. There's also good and increasing evidence on low bar dose. But one of the things that we're planning is a prospective study where we will use the gamma scintigraphy to look at two formulations with substantially different APSD, so different formulations, in order to show that the technology also has discriminatory power to show that different formulations effectively end up in different parts of the lungs. We will look at healthy volunteers and diseased patients using the gamma scintigraphy, and we'll do then the CT and the deposition in the same patient cohort. So we'll bring it all together under the SmartTrack umbrella, where we'll use the in vitro testing, the CT, the modeling, even the pharmacokinetics to kind of make sure that it's an end-to-end solution. So in conclusion, we're very happy with these initiatives around model master files because we do think it'll streamline the approval process. We do think the context of use is an important part, and I think that warrants several discussions, I think, with the agency. And I think in that sense, showing what is clinically relevant and showing that you introduce that in your models in the right way, I think will be a pathway to success. And in this case, I think making sure that the right patient-specific parameters are included is quite crucial to have what it is that we're trying to do, and in this case, having a digital equivalent of a clinical trial. So thank you so much for your attention. I'm looking forward to discussing more during the panel. Thank you very much.
All right, thank you, Dr. De Backer, for your excellent presentation. So I'd like to welcome Dr. Markus Fridén to the podium here to present his talk.
Okay, thank you very much. There's good probability that I'll be preaching a similar gospel as the previous speaker. So I'm leading the biopharmaceutics team in the Department of Inhalation Product Development at AstraZeneca, and I'll be talking today on the scope and some exploratory principles for conducting virtual comparative clinical endpoint studies with physiologically based biopharmaceutical modeling. As you will know, these clinical studies is something that you're sometimes asked to do if you're changing something about your drug product or if you're developing a generic drug. So I'll begin by saying that PBBM has a great potential for this, and the reason why I say that is that we're already using this methodology for decision-making processes in development on more complex matters than like a post-approval change. So we use this when we bridge between highly dissimilar products in different phases of clinical development. We even use it in drug discovery when we think about starting inhaled projects. Which target can we pursue with which drug modality? We take those decisions based on predictions of local pharmacodynamics, and that is without having any in vivo pharmacological data following inhalation to rely on. At the bottom there, you see a case study we did some time back where we established a PK/PD model for salbutamol using systemic exposure, fed it to a inhalation PBPK model, and predicted the time course of lung function improvement from the simplest assumptions. So this can work in my mind. So today I'll try to describe a a workflow that we are in the process of developing to support the propellant transition for our pressurized meter dose inhaler product portfolio to a low global warming potential propellant. And I'll discuss some challenges with the general challenges with establishing model credibility, and then lead on to how master model files could help us. So here's like an end-to-end visualization of the workflow that we are envisioning. At the core, you've got a CFD supported PBBM model, which is parameterized by chemical and non-clinical API data and a plethora of PK datasets. So this accepts the pharmaceutical product data, sort of batch release data, and predicts pharmacokinetics and bioequivalence. And if you add sort of additional factors to this, like study factors and population factors to the extent that is required, and also pharmacodynamic datasets, dose response studies, we think we can also extend this to simulations of the clinical comparative endpoint. So we'll look a bit closer at some aspects of this. So here's the CFD model, which we are using to sort of reverse engineer the batch specific testing data into a description of the aerosol at the exit of the mouthpiece. And this includes sort of propellant specific parameters and in vitro data sets. And this is then fed to the geometry of a human subject to simulate particle deposition in realistic conditions. Then this feeds into the bigger kind of PBBM model, which is a whole body pharmacokinetic model with a very detailed description of the lung that describes all the processes post-deposition, just as we understand them today. And yeah, as I said, the model is informed by a lot of clinical data sets. And in those data sets, there's variability in pharmacokinetics. So you can use that to kind of assign a distribution of values for some key parameters in this model. And that allows you to simulate not just a mean PK curve, but something of a realistic distribution of the PK curves. And that's sort of what takes you into the space of a virtual study. So here's an early version of this workflow in action. We're simulating a study that was done a few years ago. It's published. It was a relative bioavailability study of a triple combination, including budesonide, in 2 different propellants, actually 3. So what you have here to the left is the compendial release data. In the middle, you've got the comparative predictions of total lung and regional lung deposition. And to the bottom left or right, you have the predicted PK curves along with a predicted 90% confidence interval for a very large group of subjects. The key thing to look at here is that the AUC ratios for the test and reference products look very well predicted with this approach. Cmax is not too far off. So that this perhaps gives us some indication that we're on the right track, but it's just one study, and more importantly, it's just systemic pharmacokinetics. So how do we think about the pharmacodynamics or the local effects? So one concept that we're exploring is to use the inherent properties of the PBBM model to kind of extract comparative local bioequivalence ratios of pharmacologically active free drug, which is a state in the model. And this is something that will always depend on the region that you're interested in and on the time after the dose. But with knowledge of the disease you're looking at and an understanding of the PK/PD relationship, we think you can pretty much transfer these ratios or a realistic range of scenarios onto a dose scale where you can compare like the relative potency for your two products. And by mapping this onto like dose response studies, which you may have done, you can actually start to simulate the comparative clinical endpoint with a realistic variability. So if this can be done, it has enormous advantages, of course, including not having to pull in hundreds of patients to the study. But then the challenge is, as always, about model credibility or validation, if you will. So how do you approach this? How do you show that your model is this precise to kind of replace a bioequivalence study when actually the clinical data is often very variable for inhaled products? And maybe we have some inspiration here in this ASME VNV40 guidance that speaks to the concept of a model risk, which considers the consequence of the decision, but also the weight of the model in informing it. So the model is one part, but the other is what data. Do you have some clinical data on your product? What data do you have on highly similar products? Have you kind of studied the same or a similar problem before? So we're kind of, while this is a challenge, we're kind of considering this so almost like a weight of evidence approach, which goes kind of far beyond a numerical exercise in showing verification of your modules. It goes beyond showing predictivity of certain data sets, but it probably should consider like the complexity of what you're doing. How many properties are different? Could they be interacting? What quantity and diversity of clinical data has gone into building the model and validating the model, and so on. So this, whilst the implementations of the models is kind of doable, I think this is the real challenge, like the process of kind of showing credibility. So to the model master files, we've seen a number of cases of this over the last 2 days. So it should be quite obvious that there's many elements of this workflow that could be in scope of the master file. Maybe I should say, or just make a comment that we have a portfolio of 4 to 5 products formulated in the same platform that are in scope first for a propellant transition. But there could be other changes down the road, post-approval changes or other kind of life cycle management things coming along. So it's kind of easy to see how we could benefit from having a master file to support this. Probably my favorite one after reflecting these 2 days is kind of to have a deposit of the historical clinical data and the in vitro in vivo relationship, and maybe also some part of the thinking or the relevant parts of the workflow as such could be a useful thing to kind of have have an agreement on, or a description on, that we can sort of return to. So with that, I'd just like to acknowledge the fantastic collaboration that this has been and is being between several teams in the CMC side, and I should say clinical pharmacology. We have also collaborated with a couple of external partners on the early development of the spray. So that is Cambridge Consultants and Nanopharm. That concludes. Thank you.
All right. Thank you, Dr. Fridén, for your excellent presentation. So for our next speaker, I'd like to invite Dr. Abdullah Hamadeh to the podium here to deliver his talk.
Okay. Well, thank you. Thank you to CRCG for organizing this really excellent workshop. I've certainly learned a lot. I was expecting to fly back home with the idea of the model master file sort of crystallized in my head, and I think I'm going back with even more questions, because the more you, once you discuss the broad outlines, you start to think of the details, and even more questions emerge. So in that regard, I'd like to offer some thoughts on how dermal modeling can inform the development of this framework. The aim of the framework has been discussed, is you want to develop a structured way to communicate models, to certify their validity, so that we don't need to revisit their validity every time there's a resubmission. So to make this framework a general one, I think it's worthwhile to look at some of the intricacies that could arise in dermal modeling. And I say that because in general, there's quite a lot of detail that goes into specifying a dermal application, right? So even for the same combination of model, a compound and formulation, there could be many things that vary that induce differences in the absorption profile. So for example, the absorption, of course, is sensitive to the formulation, because you could have an aqueous formulation, an emulsion, a gel. All of these will have different impacts on the absorption. They might impact the skin differently. They might evolve differently on the skin. So whatever comes out of the tube is not the same formulation that's on the skin a few minutes or a few hours later, right? There could be evaporation of some of the components of the formulation. That could lead to two things. It could lead to a concentration of the compound on the skin surface, or if it could lead to crystallization of the compound on the skin surface. So if there's a concentration on the skin surface, that'll drive the flow, the absorption to be fast. It'll cause the absorption to become faster. If there's crystallization, then that could cause the absorption to essentially stop, right? So that's one factor to consider. In addition to that, the anatomical site to which you apply the formulation has a bearing on what's absorbed. So variations in hydration level and skin thickness will have an effect on what's absorbed. And in addition to that, the dose has a bearing here, because you should not necessarily expect there to be linear kinetics here, right? If you double the amount that's applied, you don't necessarily double the amount that's absorbed. There could be saturation effects that come into play. So this forces us to include, I think, within the model master file, a precise description of the application scenario and the assumptions that are included in that application scenario. Now, on the bright side, there is a wealth of data out there that quantifies dermal absorption for very many compounds. Even for the same compound, you'll find data of how it's absorbed from different formulations. And these can be used to validate the model. In addition to that, there are many different kinds of assays that are well-established methods to measure absorption into the skin. So you can measure percutaneous amounts, you can measure accumulation in different skin layers, you can measure the residual amount on the skin after an application. So all this is very good. So you can use it to validate the model, but it's important within the model that you define the quantities accurately that will be reflecting those measured quantities. So for example, if you have a model that says, it's simulating the epidermis accumulation. Well, what do you mean by epidermis, right? Because do you include the stratum corneum, or are you just simply referring to the viable portion of the stratum of the epidermis, right? So it's a bit intricate, and you have to sort of refer to a specific region of the skin, a specific range of depth in the skin. So this all has to be defined, I think, in the model master file. And so part of the reason why I think it's useful to look at dermal absorption is it reveals some of the nuances that might not necessarily come up in other modes of application. So in our lab, we've been active in the area of PBPK modeling of skin for some years now in collaboration with partners at Procter & Gamble and at Beiersdorf in Germany. So in that time, we've worked on extending some previously published models with some descriptions of vehicle volatility and the impact of that on absorption. We've also developed a Bayesian learning and extrapolation suite that we've used for in vitro to in vivo extrapolations. And we've explored some model-based methods for designing informative experiments with which to train these models and extrapolate them to new contexts. So recurring themes in all of the above works, and also in many of the talks that are featured in this workshop, are model building and verification, model training and validation, and then model extrapolation. And so because many of these themes come up again and again, we can sort of organize the contents, if you like, of the model master file into these three categories, sort of like the template idea that was mentioned yesterday. So the benefit of this approach, I think, is that it builds a kind of commonality, right? Almost a common language across model master files, because these elements will recur across multiple modes of applications, multiple workflows. And they tell a story about how the model was developed, how it was validated, and how it was successfully used, hopefully successfully used to predict data sets outside any training data. So I'll be repopulating this grid as I go through some of our workflows today. I'll say from the outset that within our group, we generally use the Open Systems Pharmacology Suite. As the name suggests, it's an open source software platform, freely available. It has its own dedicated GitHub and a model versioning system and a model qualification framework. So it is open source, but because there's a model versioning system, you can always cite a specific model, and everything's reproducible from there. So the suite's composed of two primary applications. So the first is PKSim, which is used to develop whole body PBPK models. And its companion application is Mowli, which is used to build custom compartments of organs or tumors, for example. It's software where you can build a mechanistic model from scratch, essentially, right? You specify the spatial structure of the model, the kinetics of transport, kinetics of the reactions, compound-specific properties. And once you have that mechanistic model in place and built, you can append it onto a whole body model from PKSim, and then you can simulate the integrated system to look at the pharmacokinetics within the organ that you're interested in. I want to add that these models in this suite, they have a modular structure. So if you open up the model, you'll find a dedicated building block which contains all the attributes of the compound, a building block that contains descriptions of the anatomy and physiology of the model, another building block that describes the kinetics of transport of reactions, and another building block even that describes the dosing. Right? So I'll come back to this modularity idea later because it is useful when you perform extrapolations. Around 5 years ago, we embarked on a project with our collaborators at Procter & Gamble to build in Mowli the University of Buffalo, University of Cincinnati casting model of skin absorption. So the model describes absorption of a compound applied to the surface of the skin as it diffuses through the various skin layers into systemic circulation or into a receptor medium if you happen to be simulating an in vitro scenario. So with this model, you can simulate the accumulation and the kinetics of an applied compound in different skin layers, and then you can, of course, use these different skin layer predictions to compare with any data, experimental data that you may have. So if you open up the model, if you look at it all at once, it might appear a bit complex, a bit overwhelming. I think if you break it down, it's not so bad. So it's essentially composed of a one-dimensional partial differential equation that's a function of partitioning and diffusivity in each of the anatomical skin layers. Now, if you take any one of these partitioning and diffusivity parameters, what you could do is you could break down the anatomical layer into different phases. So here we have the stratum corneum diffusivity. You can break down the stratum corneum into different phases, a corneocyte phase, if you like, and a protein-based phase. And then you can look at, you can start to define partitioning, diffusivity, permeability specific to each of these phases. And the overall stratum corneum diffusivity is basically an aggregate of those of those different phase-specific transport parameters. So here I'm showing on the right, bottom right, is permeability across stratum corneum lipids transversely, in the transversal direction. You can see it's a function of molecular weight. It's an empirical function of molecular weight with some uncertainty, right? And that uncertainty, of course, it migrates into your model. Once you include this parameter in there, you need to take into account the fact that it's an uncertain one. In your simulations, you can simulate that uncertainty in there. Back to the grid, what would we have here? Well, in our case, what we would do is we would reference the original publication from the literature from which we built this model, right? But it doesn't have to be, of course, from the literature. It can be a reference to another model master file or multiple model master files if you're taking bits from different model master files. It could be a reference to a particular software platform and a version number of the model within that platform. And in addition to that, what you should probably be adding is some verification steps, right? Showing that it's been this model has been built technically correctly, right? It's technically correct, right? You want to maybe include some mass balance simulations of showing mass balance. If it's a model that was derived from the literature, you want to at least show that your built model can reproduce what the original publication simulations showed, right? You want to be able to reproduce the original model's outcomes. So that's what we included in this first build and verify step. Now, once you built your model, you simulate it. It's not necessarily going to immediately agree with the observed data, in which case you need to train and validate the model, right? So what do you do first? You conduct a sensitivity analysis. You identify the most influential parameters. You take a subset of those that are uncertain, and those are essentially the ones that you need to look at optimizing. So once these parameters have been identified, what we do typically in our workflows is we resort to Bayesian MCMC method. So I'll refer to the publication on the bottom right here, where we've described this method at length. But we use this approach mainly because mechanistic models such as these, I mean, they have many parameters, possibly many of them that need optimization or training or learning from data. And what you'll find is that there is going to be some complex correlations that exist between them. So rather than running an optimization where we arrive at maybe one optimal point, what we try to do here is we try to find an approximation to the joint probability distribution of these parameters. So on the top right here, I'm showing an illustration of what the outputs of this algorithm might look like if you're trying to learn the likely values of parameters K1 and K2 in this two-dimensional space. So you can think of the intensity of the yellow contours there as being, the heights of hills or mounds that reflect the probability of the probability of a specific value in that two-dimensional space. And so what the MCMC algorithm tries to do is it starts to generate samples by looking at goodness of fit, essentially. It tries to generate samples within this two-dimensional space, where the density in this two-dimensional space of these samples is essentially reflective of how likely a particular point is. So where you have high density of samples, you're likely to have a high probability that those are values of the parameters K1, K2 that will give you a good fit to the model. So we end up with this sort of kind of two-dimensional histogram that tells us where the most likely values of these parameters are. To show you what the output of this algorithm looks like in practice, so I'm showing here with this slide, the fits of the model to testosterone skin absorption measurements from the 2019 Cosmetics Europe study. Here, the visual predictive checks on the top left show the fit of the model to different to testosterone accumulation measurements in different model compartments, whereas on the top right, you have fits of the model to percutaneous testosterone accumulation, percutaneous absorption amounts pertaining to different individuals, right? Because for testosterone, every individual, the compound was tested on 4 different individuals in triplicate. So the MCMC algorithm, what did it do here? It was able to integrate a data set that includes both data from different model compartments and from different individuals. And it was able to learn a joint distribution between compound-specific parameters and individual-specific parameters. And I say joint distribution here because, remember, there are going to be correlations. You can think of the permeability and the skin thickness as being correlated here. So the learned marginal distribution of testosterone on the bottom left, I'm showing in red. Well, what can you do with that? You can use it to simulate testosterone absorption in other contexts. Whereas the marginal distributions of individual-specific parameters, which I'm showing on the bottom right, those you can use as a way of quantifying inter-individual variability within this small test population, right? So you can, if you ever want to simulate inter-individual variability between in this small population, you can sample from these individual-specific histograms. So to move back to the grid, what would we put here? Well, you would put, I think, the results, the histograms, the training data that you used, perhaps the sensitivity analysis, visual predictive checks. And why do I say we add the histograms and the training data? Because you can always use these. This is useful information, right? You can use it, first of all, to validate the model, but you can also use it for extrapolating the model into new scenarios. Now, moving on to this final extrapolation stage, we want to show that the trained model is able to extrapolate and predict data sets outside the training data, right? So here we can take advantage of the modularity of these models. Essentially, what is extrapolation here? Extrapolation, for example, to a new formulation means that you simply take the formulation-specific model building block of the model and you update it to reflect your new formulation's attributes. You could also simulate in vivo skin by simply updating the building block corresponding to in vivo skin, or you can simulate a different dose to the one that you use to train the model. So in this slide, I'm showing how the learned distribution of testosterone-specific parameters were used in extrapolating the model the trained model, I should say, to predict dermal disposition under the in vivo scenario. So here, the building block pertains to skin was updated to reflect in vivo skin. And then we reused the distribution of testosterone-specific parameters to simulate the model, which gave us a range of predicted testosterone absorption amounts. So what would we add in this third stage? Well, we would add information such as what modules were updated, what data we're trying to predict, and then the prediction, the simulated testosterone accumulation under this new in vivo context. So I'll conclude here. I've presented some ideas about how the contents of the model master file could be structured. We want, at the end of the day, to say how it was built, how it was validated, how well it does in predicting. I think, the dermal modeling example can, I think, show us some of the nuances that come with building a model master file. The workflow might be slightly different to what you might find with other modes of application. And I did highlight this three sort of category way of compartmentalizing the model master file. I think the really interesting outcomes start to happen when you start to get two model master files and start to take what you've learned from each one and putting those together. So imagine you have a virtual bioequivalence workflow where you have a reference product. You can build a model master file for that product. Maybe you can learn some of the inter-individual variability in their target population from data from an in vivo study for the reference product. And then you can build a model master file for the test product, right? But you don't have an in vivo study there, so you might use in vitro data. So then what you would do is you would maybe simulate the reference model that you've developed. You'll get simulations of disposition for the reference product. Then you can take what you've learned from the test product's model master file and use that to extrapolate the reference model, and then arrive at simulations for the test product. And then you can use the simulations for the test and the reference to conduct your calculations of virtual bioequivalence statistics. So I think there's a lot of possibilities when you start to combine multiple model master files. A lot of interesting things can start to happen. I'll stop there and we'll meet at the panel. Okay.
Thank you, Dr. Hamadeh, for your presentation. So for our next speaker and our final speaker for this session, I'd like to welcome Dr. James Clarke to the podium.
Hi, my name's James Clarke, and it's a real pleasure to be here. I want to start by maybe telling you a story about the build-up to preparing for this workshop. So I have two colleagues here, Nikunj, who spoke yesterday, and Amin, who's going to be presenting in the afternoon. And when we realized that we were all speaking here, we decided to meet internally and have a discussion. And it turned out that none of us had any clue what a model master file was. And all three of us actually had very different ideas of what that might be. So that was quite interesting trying to prepare for this presentation. And I think we will all present our different views of what that is. But having said that, being the last speaker today in this two-day workshop, I think that I'm in a position now where a lot of the questions that I've put in these slides that I'm about to present have been answered over the last 2 days. And I have a much better idea, at least, of what the MMF is and how it might work. So I think that's really good. And as I go, actually, some of the questions that I've put on the slides, I'll try and answer them, I think, as to my understanding now that we've discussed them. So the first thing I wanted to focus on was a kind of what resolution will an MMF be submitted. So there's lots of different types of MMF potentially that we've discussed, and the level of detail that's required, and what might be included in that. And I'm also going to look at a couple of use cases. I'm going to use real-world examples of submissions that we've done in the dermal absorption space to discuss that. So being a PBPK modeler, I'm going to start from the bottom up. So the most kind of zoomed in part of the MMF, I think, is the use case. So here I've used the example of a safe space analysis as a use case that you might have here. And this is the context of use that we've been discussing. So each MMF will need to define a context of use. It doesn't necessarily need to only define one context of use. It can be a range of use cases, but I think that probably needs to be quite well defined in the MMF. So the next step above that is kind of in the context of the safe space analysis would be a parameter that we want to do that safer space analysis on. This is quite a specific thing, and so we wouldn't be submitting an MMF just for this specific. We need more detail of what lies above that. So we have a mathematical model in which that parameter fits. And this is the kind of range that I think we want to be focusing on for submitting an MMF, is focusing on a specific mathematical model. Here I've used the example of an emulsion model in which a droplet size parameter fits, where we might do a safe space analysis on that parameter. A step above that is a module. So I've used the example of the MPML-MEC derma, which is the dermal absorption model in SimSip here. But I think we're getting to the range now where an MMF for a module this size is maybe not the approach that, well, at least in my opinion, that we'd be thinking of, to have one MMF just defining that whole model. I think that maybe the best way to describe that whole model might be a series of MMFs that describe different use cases. So I think it would be possible to have an MMF that describes, for example, the MPML-MEC derma, but it would be pretty large and would define lots of different use cases. So it could potentially be broken up. And then the final is the platform. So we've discussed having kind of an MMF that could describe a platform qualification, for example, but obviously at the level of the whole software, that isn't the kind of resolution that we would be looking at. So I think that the resolution of the individual mathematical model is the kind of specificity that we'd be looking at for submitting an MMF, in my opinion. So thinking about what we might include in that, I've kept with the example here of just an emulsion formulation safe space analysis. So we would be describing the model and its features, and this is something that has come up quite a few times as to what level of detail is going to be required, and I'm still not clear on that. That is really something that I'm not clear on, as to what level of detail is required. There's been discussion about, for example, that it might require submitting code, or that it might describe, the other extreme is just a description of the model. I think probably somewhere in between, defining the mathematical equations, but to the level of code is probably not feasible in the cases for some of these big models, and the regulators probably don't have the resource to be constantly reevaluating code at that level anyway. And a series of case studies which can consist of different categories, and this is a bit of a sticking point, I think, for how this might work technically. So published works are quite an easy one, I think. So obviously, if a case study has been published, then we can reference that. Does that then need to be included in the MMF as well, or can we just reference that in the MMF if it has been peer reviewed? I think that's come up a few times already. The answer to that isn't quite clear, whether the peer review process is sufficient or whether that will need to be redescribed and reassessed in the MMF. There's also like non-published works, in progress works, which would be included. So, for example, we have ongoing case studies that we're building as part of our FDA grants, which we haven't published yet. But if we were submitting an MMF, we would include those case studies probably. But then the final one is non-published and proprietary. So if we were submitting this MMF with a sponsor, they might have data that we can develop case studies for potentially other compounds and formulations that we might want to include in that MMF. But this creates an issue because if you submit that and it becomes an MMF, then that creates a conflict of interest then if anybody else wants to reference that MMF because it contained that proprietary data. And I'm still not clear on exactly how that could be dealt with. And there probably needs to be a way to try and separate them and be able to reference an MMF and segment part of it. But I don't know exactly how that would work. So I'm going to come on to this on the next slide. So there's not every model requires an MMF. So we frequently use our PBPK models to make internal decisions with our clients, and things like dose selection, power analysis, simulating an IVPT study before it's been conducted to look to see if the protocol is appropriate. These are things that we do to inform internal decision making, and I don't think that they require an MMF to be developed for us to continue doing those things. But I want to look at a couple of examples of where an MMF may be required, and in fact, these are real-world examples that have occurred and we've heard about already. So to replace a clinical endpoint study, we heard the example of the diclofenac gel, where a clinical endpoint study was replaced by modeling and simulation. That same example actually was used, the second example, which is to expand to local concentration assessment. So in that example, plasma data was used to verify the model, and it was extrapolated to synovial fluid concentration. So that's another example where if that was being done again and we were in a place where model master files were being submitted, then I want to just think through about how we might do that. And another example, which is published that models have been used, is to expand an age range, for example. So I'll quickly look at that example. So for the replacing a clinical endpoint study, using the example of the diclofenac gel, perhaps, for that submission, we submitted what we call a verification package, which I think Ross showed on his slide a few presentations ago, which had multiple formulations and compounds included in it to verify the platform. So if we were doing that again, if there was another submission very similar to that now, then we might think to submit an MMF in place of that verification package. They're very similar things. So I just wanted to think through maybe what we still don't know if we wanted to submit that tomorrow, like what questions do we still not know? And that's really how detailed does it need to be, which we discussed. At what level do you need to describe these models? Is it okay to provide them at the level of a SimCip workspace, or do we need to go deeper than that and describe the underlying models in significant detail? And then the outcome of the MMF might be to have a scope of acceptable usage of that MMF. And I want to just a few open questions that we have about that is the case studies that are included for this have a certain range of physicochemical properties and actually have a certain range of formulation. And it's I guess, unclear how far outside of that range that it might be possible to extrapolate. But this has become more clear to me actually during this workshop, that potentially the MMF itself, I think on the first day it was said, the MMF itself wouldn't be accepted or not accepted. The MMF would exist with a certain range of verification. So as far as I understand, the way that this might be addressed now is the MMF would exist in the public information. It would describe what physicochemical data was used to verify that. And then anybody wanting to reference that MMF can look at what was used to verify it and decide whether what their application is might be appropriate for that. So the acceptability of the MMF would be on a case-by-case basis. So I think we've kind of figured that one out. Over the last 2 days, that's become more clear to me, at least. But yeah, there's always a balancing act between kind of the specificity and the reusability. So if we get too specific and we ask to be the same physicochemical properties in the same formulation, that MMF then becomes not reusable. So there has to be kind of a balancing act between those two. Again, similar questions for expanding to local concentration, the how detailed does that need to be? The example I gave for the diclofenac gel was extrapolating to the synovial fluid. But then the question is, how many case studies might be required to do that for a different compound? Let's say that there was a different compound that was, the site of action was synovial fluid. Would that diclofenac case example serve as verification to extrapolate to a different compound? And if not, how many case studies would you need to be able to do that without actually going to measure the synovial fluid concentrations in your compound as verification? So these are still, I think, open questions, but again, it's to do with the acceptability of the model. And the final example was to extend the applicable age range. So another kind of different thing that, and a different example was an example where a PBB model was used on the label to expand the age range of a product down to further into the pediatric range because the sponsors weren't able to recruit patients of that age. So the PBBK model was used to extrapolate down into a lower age range and allow that to the label to have a wider range. So again, I think that this could be something that you could then submit an MMF for, which would allow only the pediatric dermal physiology to only have to be assessed once. And then I think the outcome of that might be an applicable age range in which you're able to extrapolate based on that information. But again, the same questions are to do with how similar the compounds need to be, because for most of these examples, we only have 1 or 2 case examples. So if we're too strict on how similar they need to be physicochemically or formulation, then it means that those MMFs are then not reusable. The final thing that this is my final slide, I wanted to discuss, and I think wanted to maybe discuss in the panel if we can, is regarding updates to the MMF. This has come up a few times, but I think it's a really important question because using the example of the SimSip simulator, which is released every year, there's a new version that comes out every year. If we are going to develop an MMF for something, it's likely going to take a while to do all of that work in the new version. So by the time the MMF is accepted, probably the next version of the simulator will already have come out, right? So we're then in a point where we have an MMF that's been accepted for something great, but we've already got this new version. We may be close to releasing another version. In some areas, this might not be a big problem. If it was something relatively simple, like a systemic distribution model, those equations and things aren't going to change in the simulator version to version. But if we're talking about dermal absorption formulation, we've got three ongoing grants. Every year, we drastically update our dermal absorption models because we're in really like heavy development for that. So the question then is, what do we do? Because there's been discussion about that previously, that this could stifle innovation. So at that point, if you have an approved MMF, that really creates a kind of potential stifling of innovation because it gives the incentive not to change your model because you'll have to go through the MMF process again, which isn't ideal because we want to be updating these models to keep them right at the cutting edge. So we need to figure out a way to deal with that. I think at least creating a relatively simple pathway where we can update the MMFs when we do create new models without having to redo the whole thing would be really useful to try and reduce that burden both on the resubmission, but also so that we don't stifle innovation in actually updating these models as frequently as we can. So I think that's all from me. Thank you.
I thought there was another step. Okay, welcome back. It's always challenging after a lunch session, but we have a very esteemed panel here. So let me start by introducing myself. My name is Sujatha Sonti, and I lead the drug product development at GSK, a team that is responsible for developing all dosage forms, oral, inhaled, sterile. We have a process engineering and device engineering team as well, and routinely, we use all the different models that we've talked here. Additionally, as a company, we've developed a number of CMC-related models as well. So this topic is extremely dear to my heart. So in the interest of time and having seen the amount of questions we've had and discussions, we have decided not to spend time introducing each one of you. But if that's okay, when you answer questions, if you could introduce yourselves, that would be fantastic. Excellent. So the way we're going to do is we have a few questions that are already pre-submitted. We have that. We will obviously take questions from the audience. And Alam is going to help us also moderate questions online. So that's going to be the format for today. So maybe I'll start with the first question that was more specific to local administration, because this was a topic of discussion for this panel. And maybe Ross and can take the first question. So when modeling topical drug administration for local action, one cannot directly presume that validation with systemic PK validates the local exposure predictions at target sites. So what viable approaches can one think of to validate predictions for local concentrations? So I'm just going to direct it to you, Ross, and then anyone else can join in.
Hi, Ross Walenga, FDA. So that's a great question, and for a lot of it will depend on the product that you're talking about. Is it orally inhaled, ophthalmic, applied on the skin, or some other route of administration? But for orally inhaled drug products, if the point of your model is to predict regional deposition, well, there are in vivo techniques for that. There are imaging modalities, gamma scintigraphy, SPECT-CT, even PET-CT is a new emerging technique that you can use to directly compare your predictions. But then again, it may depend on the type of drug that you've got. So say, for example, you have fluticasone propionate, which is a slowly dissolving molecule. Well, then maybe deposition isn't everything you need to know. Maybe you need to know more about the absorption. In that case, it could be a little bit more complicated. I think the way we've talked about this in the past is using kind of a bracketing method where you're validating the deposition and then you're validating systemic PK. So you're validating upstream and downstream, but still you're not really getting at the local tissue. I mean, there are some limited studies using like bronchiolar lavage to actually get local measurements, but these are quite rare and quite invasive, so not something you would do routinely. So that's how we've approached it for that. I think for ophthalmic products, and turn it over here to Mingliang.
Sure, yeah. Thank you for the very interesting question. This is actually a really challenging area for us for the locally acting product. For example, for the ophthalmic product, the products are usually applied to eyes directly, and the APIs reach the site of action, usually the local eye tissues, before reaching out to the systemic circulations. And also for the systemic part, usually it produce very low to undetectable plasma concentration. So, but fortunately, for some product, we still have some way of validating your local tissue prediction. For example, for some ophthalmic products that are indicated for cataract patient undergoing surgery, so you can have some PBPK model by considering the drug release and the drug permeation across different eye tissues, and then using the aqueous humor, for example, data from cataract patient to validate your whole PBPK model. That's one example. Another example, I would say, for example, for the ocular, you could build some ocular PK-PD models where you use local tissue exposure data. You predicted to build your PD data, where PD may be measurable, for example, for some products that indicated for lowering the intraocular pressure, like IOP product, where you can directly measure the changes of intraocular pressure so that you can indirectly validate your local tissue present, like the predictions. That's another example. So again, it's a challenge area, but there's still some possible ways of doing some direct or indirect prediction or the validation. Thank you.
And I'm thinking perhaps in the dermal space, and maybe James, you or Eleftheria can help as well. You can use techniques such as microdialysis, perhaps, to measure local PK. Is that?
Yeah, I mean, there's some in vivo techniques. The data's a bit more few and far between in vivo. There's IVPT, local concentrations. It's still unclear how correlated they are to in vivo local concentrations. But the idea of the mechanistic models is that we can predict the relationship between the local concentrations and the plasma. All of the layers have QSARs that predict the partitioning behavior between them from physicochemical properties. So, and this comes back to something I said in my talk, is if we can develop a number of case studies that show that the model with its base predictions from QSARs from PhysChem is able to get that relationship right between local concentrations and plasma, then we should have some confidence in predicting for another molecule that if we have plasma data to verify, we can predict the local concentration without going to measure it. That would be the aim. I don't know if we're quite there yet, but that would be the aim really. And if we could submit, in this case in MMF, to show that we can predict local concentrations that can be referenced, then that's the ultimate goal, I guess.
Markus, yeah.
Yeah, I'd just like to build on these things. Like, I think regardless of the administration route for local treatment, there's always technical challenges in the experimental measurements that one needs to consider. But I think the process of comparing your predictions to whatever you can measure to some extent is sort of part of the building the credibility. So I think all these approaches are useful to always try to fit map the model onto the data that are accessible to kind of build confidence in the local predictions.
Okay, thank you.
I think if
Yeah, go ahead, please.
Yeah, I think if you look at some of the sunscreen literature, in vivo studies, so they conducted a study where they applied sunscreen to like 75% of the body, and then they monitored plasma concentrations, but they also monitored the accumulation in the stratum corneum, at least the upper echelon of the stratum corneum, through tape stripping. So I think one can arrive at a strategy where if you have an in vitro model that you've validated, so you know something about the diffusion across the viable layers, and you also have data from tape stripping experiments and plasma, then you can arrive at predictions of what's going on in between the stratum corneum and plasma, basically.
Hi, Jay Mowli from Capstone Development. I think there's a lot of different techniques in the literature, like in vitro, in vitro permeation, microdialysis, as you mentioned, DOFM, in vivo tape stripping. I think as sort of a sponsor and interacting with maybe a CRO that's for, for example, like dermal PBPK modeling, it's really important to understand what's the underlying data. And I see like huge value, kind of as James mentioned, in like MMF for secondary verification or like platform verification. I think it's unlikely that MMF would have exactly maybe the context of use or the compound that maybe a sponsor would be, especially like a generic sponsor would be going primarily to address. But in the secondary verification, there's kind of it's a huge, I guess, scope that could be addressed in advance in the MMF. And knowing what's the underlying data for the platform verification can help guide understanding or having a mutual understanding with the collaborator, what data needs to be generated as part of a model-informed submission.
So just so I understand, what you're suggesting is a platform, the verification of the platform in itself is very helpful. In the context of use, then becomes a discussion.
Yeah, and sort of, having, as James mentioned, a series of case studies over a certain physical chemical space to kind of, because I think like in terms of the exact compound and formulation, that's included in the application, a lot of that will be proprietary information. So maybe the compound file may exist, or different formulations can be used as part of this verification process, and maybe that's where there's significant use for the MMF concept.
That's a great. Yes, please.
Yes, I think there are two levels of discussion here. There is, on the one hand, because I hear about proprietary nature of some of the information, and I also hear about platform, what we call verification and validation can be subject to debate, but we understand that these are the data we use for credibility establishment of what we're trying to include to support the MMF. On the credibility establishment, so to say, I wouldn't put the intellectual property as a barrier, because I think at some point the regulators need to assess the validity of the platform. They have access to that if we are working under a confidentiality agreement. We can see, what is not disclosed. So there's one level which is what we establish, the validity, the credibility of the tool. On that, I wouldn't really care about intellectual properties. Now, when you have something which is kind of validated, you have delimited the space for applicability of the tool. Now comes, what do you want to share? This is where now the debate on, is it going public? To whom can I show or can I not show? But the regulator should not be part of that debate, actually, because we would see that even if it is resubmitted 10 times. Now, when comes like you have delimited what you want to share or not, to whom the modern master file can be used or not, then we can now start thinking about if I have to share to the competitor, do I want him to see this and that is confidential. So that these are two different levels. And when we discuss, I think that it is really important that we disentangle which level are we discussing. Thanks.
Thank you. I'm just going to continue on this phase of discussion, just for, if that's okay, Alam. One of the questions which is very relevant is a question around, it's a more general question around MMF. Very often an MMF may require diverse data sets. So for example, in a dermal, you may need in vitro and in vivo modeling. So the question is, do you envision all of that data being a part of a single MMF representing both? Or could they be, do you see those as two separate MMFs? I welcome that discussion from anyone in the audience as well.
I mean, as you used the example of dermal with in vitro and in vivo, I'll take it first. I guess it kind of depends on the use. There's a lot of the time we're looking to do in vitro and in vitro to in vivo extrapolation, whether that be from IVPT data, but even if there's not IVPT data, we're still doing IVIVE because we're taking in vitro formulation parameters and things and extrapolating. So I think they have to both be there because most of on the PBPK side at least, that's not true maybe in some of the areas, but on the PBPK side, we're always doing IVIVE to some extent. So it will include in vitro and in vivo data, I think.
Any thoughts from the audience? Yes, I think there's a question. It's a separate question. So do you see it almost like an inheritance model? Because that was another kind of a question that came in, is you have an in vitro, in vivo, but they're linked towards the same. Is that how you would envision it?
Yeah, I think so. I think if you can split up the model into different building blocks, right?
Yeah.
Where you learn all you can out of all the data that you have from one, and then you inherit it. And you can be inheriting, for example, from multiple MMFs, right? I mean, if you have a well-characterized formulation and how its impact is, if you have quantified its impact on skin from one MMF, and then you have another MMF that tells you something about the compound, you can bring those in together into a new model for sure.
Yeah.
Yeah, I was just going to say, I guess I would also ask as well, what would be the practical distinction, between having a single MMF that it combines the in vitro and in vivo model versus two separate MMFs? And that's something that I think may be different. Maybe I'm sending this off to a CRO, and they do these all the time, and they have their own model of in vitro, and I put my model of in vivo on top of it. In that type of situation, you might have their MMF, that you grab, and then you have your own MMF, for example. But if you're doing everything in-house, you may put it into a single file. So I'm not sure, there may not be a single answer to this question.
Yeah, I think in the inhaled space, it's very similar to that, right? So the input parameter for any modeling is usually the APSD and the inhalation profiling. I think in the MMF, you can specify what the ideal input parameter is. For instance, if your whole thing is built around as much patient-specific information as possible, then it makes sense to measure the inhalation profiles in the target patient population, which is substantially easier than doing a whole clinical endpoint study with FEV1, of course. So I think there's a lot of value in combining in vitro, in vivo, and in silico, which adds, I think, to the credibility of the approach in general. And I think that the model master file could be a good guide to which elements can you take from where. And to your point, it doesn't have to be all in-house. It could be referencing other model master files where needed.
I wanted to add, thinking about the topical drug products. So if you had a model for IVPT delivery or an in vivo model, I guess I could think of one reason why you might want to have separate MMFs is if you have a model where you're expecting to make significant updates to one portion of it, but maybe not the other, and then you want to avoid kind of future changes to the MMF. So if you've got your IVPT model and you think that that's great, you're not going to change it, you might want to have just a separate MMF for that because you're not going to have to make changes later. And then just make changes to the in vivo model separately.
We should probably go to
Yeah,
questions.
may
from
I
audience.
add here something? So submission of MMF doesn't require that it has to be reviewed by FDA right away, right? So it will be reviewed once it is referenced in applications. So that means that there is nothing stopping you to submit an MMF only for in vitro or in vivo. It will be the review issue when your MMF is referenced in an application and applicant want to use your model for the local tissue prediction or local concentration bioavailability prediction. So that time probably you need to reference different part of different MMF in your current MMF, for example, and every will review that whether it is suitable or not.
You have a question, I think you need, yeah.
Maybe it's related. So, Andy Hooker from Uppsala University. So, you sort of referred to this idea that the MMF is only going to be reviewed as it's coming into a submission. But then if you have a platform that you want to have as an MMF or some sort of methodology or a procedure that you want to have as an MMF, then it's larger than just one submission, right? And so to me, that's going to take more effort. And so I wanted to actually see what Flora thought about that, because you have these, I think it was 7 or 8 of these submissions last year. I can imagine it takes a lot of time it's extra time, right? It's not the time for a standard submission to look at those 7 submissions per year. And do you see that you're gaining? So I can see the advantage of it's furthering the science. It's making scientific advances by saying these procedures are better than what was previously used. But just in terms of efficiencies, are you seeing efficiencies, or are you seeing that you're spending more time on these qualification procedures and not getting anything back, or getting little back?
Actually, I understand what you want to make as a point, and I agree, actually, that we are only interested, but this is where I think the MMF might be slightly different from the qualification procedure. Because in the qualification procedure, by aim, we are only interested into working on innovative method that are intended to be actually used in well-identified drug development program. This is why I showed you one example where we had that academic, a team that came to us like they go for a publication, just to have a sort of stamp on something that is not intended to be used in that form in future drug development. We stopped it very short. We're not going very long, lengthy discussion, investing our resources on something that will not end up being used in drug development program. So we will prioritize the ones for which the drug development program, the future client, are well identified already. And most of the time, these potential clients are part of the applicant team, actually. And this is the way we see it. And the outcome of a qualification procedure is an advice or an opinion. The way I understanding from our FDA friends, and please correct me if I'm wrong, is that this is open. At the time you submit the MMF, it is not accepted for the future application yet. This is where it's different. All the procedure that I have qualified, I will tell you exactly how I envisage them to be used. If something changed, I want another qualification, because this is not what we agreed. Here, they have some more flexibility. This is where, you also have some margin for what can you include just in case, never know, someone might find that useful. We don't have that one. And I agree, we don't have the resources to effort that one.
Yes, please.
Isn't it also a little bit of a question about what comes first? I would assume that, in our case, we would go down the model master file route with an approach that already had some regulatory success for a specific product. So if that's the case, then you would assume that that part is, let's say, good enough to be put into a master file process of some sorts. And then you can build from there and see, you would assume if it's successful for one product, specifically for complex generics, there's other products that are specifically there, it could also be successful. And then you build from there. I don't think you would start out with a model master file and then hope there'll be a regulatory success down the line, because that doesn't seem to be a very efficient process.
Really good point.
I'd like to add to that. One thought along those lines is that right now, as far as I know, at least in the Office of Generic Drugs, we don't have a pathway for meeting with a CRO before they would submit an MMF, or for anything, really, unless they're tied to an application. We have pre-ENDA meeting, we have the new MIE pilot, but it still needs to be tied to an application. So if there were to be some sort of meeting pathway to meet with CROs, would then there be some incentive to submit an MMF before it's included with an application?
And the question is, will it actually make it more efficient in certain cases? We talked about that yesterday, is if there are models that you routinely use, and it will help the agencies to reduce the amount of times they have to review, is this something that the agency should perhaps seek out certain CROs that they routinely use and get MMFs on those as platforms? And is that, it's something like a mix between what you're doing and what it is here, which might be something for the agency to consider. Sorry, somebody was trying to say something here on the panel. Yes, please.
Yeah, maybe I can add one point to your original question. I do understand, right now, for example, even for right now, we don't have MMF approach, right now, mechanism. Even in this case, for all the models which are submitted with the application, so regulatory agency, for example, FDA, does need to review that, like the model, everything extensively anyway. So once, for example, after this reviewing for this package, and the model is acceptable, then there is a potential, we do say the beauty of the model master file potentially in the future, like any other applicant or sponsors, they can reference that. And also for our regulatory agencies, we do see mitigation of review burden for the whole process. So I still say, I think that's one beauty of the model master file. Even for the new models, we have never reviewed. For the potential future use, that's would be helpful, I think.
So maybe you can nudge them if it's a really good model to do that for the next time around, especially if it has wide applications, perhaps, because you have to start that process.
I think maybe one bit of a difference between qualification and MMF as well, as far as I've understood, is there is some scope for like off-label use maybe of the MMF, where, I think of the example I used where you might have verified with a certain range of phys-chem, but you could probably submit for something outside of that range and give sufficient kind of evidence that you believe that's acceptable, and then the FDA will assess that, whereas I think the qualification process doesn't allow that off-label use, right?
No, there's some nuance in there, because it depends. The good example for that is imagine one CROs or one consortium is developing a disease progression model, and then they are implementing that disease progression model in a clinical trial simulator. If they come for qualification for the clinical trial simulator in a particular setting, we will give them qualification. The simulator is qualified only for the different scenarios, space of scenarios that were included in the qualification briefing. Now imagine that they don't come and qualify the simulator, but they qualify the disease model. And that disease model can be used in different kinds of clinical trial simulation. It's actually the way they frame it. This is why we discuss the context of use. When we see that it is like the virtual population that is problematic, we will advise them, restrict your context of use. Don't go for qualifying the simulator, qualify only the disease model. It gives you flexibility. If in the qualification opinion statement we say that this disease model can eventually be used for different type of application that request a disease model, it also gives you some sort of possibility, broader space, so to speak, for using the model. So it depends, but what we are qualifying need to be clear. I agree that the part that is part of the qualification story, that one is static. Yeah.
But if you have to add use cases to it, it probably is going to be simpler if you've already qualified the base model.
Exactly, and this is what we
Yeah.
always advise, but I don't know why the applicant always have the perception that the wider, the broader context of use, the best. Personally, as a regulator, people think that this is because of the work burden, but no. I think the best strategy is to have a restrict context of use. Actually, it gives you flexibility. One speaker yesterday was telling us about the life cycle of the model. I mean, the more restricted is your context of use, the more direction you can give it after. If you have a very broad one, for that one, of course, it's ready for being used. But then you have much less of flexibility outside the qualification set. So it's a strategic discussion here, actually, depending also on the applicant.
I think it's a very important point. I think what you mentioned is that you need to have these interactions with the agency to do it. And to Ross's point, currently there's not a lot of opportunity for that. So maybe one of the takeaways could be that maybe as part of the Quantitative Medicine Center of Excellence that was just established, maybe there could be some sort of framework where we could discuss exactly that. Is it better to do a broad or a small context of use? What is required? Because otherwise, if we have to tie it to an ANDA meeting, first of all, then it's up to the pharma companies whether or not they want to do that. And if they take the lead, they're not usually the modeling experts. So you're, again, dependent on whether or not you're on the agenda. So I think a separate communication channel would be probably quite useful.
So we are running out of time a little bit. So I want to check that whether we have any question from the in-person audience.
There's a question here.
Yeah, can I ask about the orally, the inhaled models? How confidence do we have in these models? And are they specific to a specific, because some of the inhaled agents modify some of the factors included in the model, like the expiratory volume or the peak flow. And so, I mean, how these are included in the model, and while they are still specific to a specific. So my question is, what are these models specific to specific drugs, or they can be used with any agents like anti-inflammatory?
I can maybe try to speak to that. I think the inhalation models for all orally inhaled drugs are that I've seen are sort of based on the same principles. There's scope to account for those things that you say, inhalation flow rates and so on. So I think the basic structure is all the same. But I think as with any type of modeling, like the validation is kind of tied to an applicability domain. It's kind of inevitable that sort of one version of the model can perform better for a certain class of inhaled products where some processes are more important than others. So I think it's really important to not treat all inhaled products as the same. And it's the same in chemometrics or any other discipline, that you have a global model which tells the big story, and then you can have highly predictive local models that are can be built on more detailed and more extensive data.
Right, I think for the inhaled part, it's really the device, the inhalation profile, the particle size distribution, and then the disease lung that drives deposition. So if you include those elements, of course, the challenge is they can be very variable. I mean, a patient can inhale very fast, very quick, or very slow, which will really change the deposition pattern. So it comes down to quantifying your uncertainty. pretty much. Making sure that if, as I discussed, if you want to have a digital equivalent of a clinical trial, you have to have a good understanding of what a clinical trial is and what are the key drivers for variability in that trial. And then you have to show that in your in silico approach, you cover that to the best of your abilities. And I think nowadays with the technology, you can do much more than maybe 5-10 years ago. So that's the good part, but you're absolutely right. There's a lot of variability in there, and it's up to the in silico modelers to make sure that you reflect the variability that you could reasonably expect to encounter in a clinical situation.
So the better the discussion is, the tougher it is to close. But I am getting some signals here that I should close this discussion. Obviously, we have some more small group discussions. I would like to now call Dr. Liang Zhao to come in and make some final words for the session. Thank you so much for the panel. And thank you for giving me this opportunity.
Yeah, just want to congratulate all. I know it's been a long day. Yesterday we started from 8 and ended at 5:30. And I guess everyone have enjoyed a dinner. Now we have another exhaustive day, I understand. Thanks for staying with us. And also thanks everyone in the online for the online participation as well. So I'm probably not going to follow my slides. I have made some, but I just want to have 2 minutes sharing on what so far we have learned. In my assessment, I think, first of all, we kind of what we have achieved. We have been making friends, first of all, that's the thing. We are actually from different distinctive groups. So for me, I'm like a pharmacometrician by training. I know we have statisticians, we have biopharmaceutical scientists, we have PBPK modelers. Even in the PBPK modeling field, we have 2 distinct groups. One in the biopharm, the other is more like in the clinical side. So I think this probably one workshop have gathered everyone together. So we may be attending APS meeting, maybe attending ASBT, we may be attending ACOP. Usually we do not gather like this. I hope we find each other lovely, we like each other. That's the first achievement. Second achievement is that we have kind of a clash of thoughts from all angles. For example, people talking about model V&V, verification, validation. So as pharmacometrician myself, initially I also don't understand the verification part. So only when I talked with the CFD scientists like Yang and Markus, then also with our internal group, I kind of appreciation where the verification comes from, is that with hundreds of equations, you need, first of all, you need to make the balance, mass balance correct, everything add up to 100%. Sometimes we do kind of wrong mathematical equations if it's too complex. Secondly, even you have written the correct mathematical equation, the computation of the mesh size or the steps you are taking can lead to different simulation results, the verification. So for us, pharmacometricians, we've been dealing with population PK models, explorer response models. We may have lack of precision verification, model verification. Now, with the group, now we all have kind of a mutual understanding, have a clash kind of a mind. Third thing, for model master file, I think what we have achieved for many of us, including myself, I think many of us have zero or minimal understanding of the concept of model master file. After one day, my exhaustive day and a half, I believe, including myself, we have much deeper appreciation of model master file. From actually James' presentation in the morning, I believe he kind of develop a slide either yesterday or today. So that reflects very deep understanding. I'm really impressed on the understanding model master file. There are many key elements that's being reflected by the presentation. I think for me, also kind of been appreciating the different feedback in terms of the nuances that lead to the third achievement. So the understanding model master file. Now we kind of know there could be thinkings from a new drug, from generic drug, and also, we appreciate, there are different types of model master files. In today's presentation, we have like a digital twin, could be type of a master file. Then we have workflow. We have thermal, like a PBPK, the process, also the things being discussed on the panel. Those can be master files. Also from, I think, from one of the presentation, either James or others, there are different, not on top of the tabs, there could be different hierarchies of master file. For example, the platform, the module, the model itself, and the parameter, the data validation, the data and the modeling assumptions, all of this have been put on the table. We later on will have a deep dive on all of the earnings later on. Fourth, I think we also have learned lots of ideas in terms of implementation, execution. So we want to, keep everyone's thought and proposals in mind. So here's what will be the future action. So I know everyone has maybe his first time attending such an exhaustive workshop. That's why we need some excitement. So we also need a payback of your effort. We're gonna summarize the learnings, comment on the proposals we have received. We're gonna conduct further landscape and scenario analysis. Learn those things from FDA. When we want to do something systematically, we need to conduct those type of analysis. I hate to use the word SWOT analysis, but I think we need to learn the benefit and the downside, potential unintended consequences, and also opportunities, and also potential, I hate to use the word threat, but kind of want to put things in the context to make model master file beneficial to everyone. Then we will develop a list of options and implementation strategies. Then probably we can, in the short term, set up a pilot program to have further real-world learning. After that, we are going to, that's going to be accompanied with outreach, education, training. May need to gather everyone's help again in the near future in terms of webinar and workshops. Really happy to collaborate again. Then after the pilot, we will probably see how we can further optimize the program, make it really not a bureaucratic, but very, very beneficial and very, very innovative, becoming a very effective tool to modernize the drug development and also cut the cost for regulatory communication, make it useful. That's the plan, action plan forward. So at the end, I just want to thank the CRCG team and the workshop organizers. Many people have contributed to the organization. You see, we are more like giving a show, not Broadway, but so many players being rotating on the stage. Thank you all of your efforts, speakers, panelists, and participants. Again, maybe it's the most exhaustive workshop you have attended, but your effort will be realized in our further actions. If we have a pilot, anything, you will hopefully will feel your own flavor in it. So we're with that, we are going to close the online session, and we are looking forward for the small discussion on day 2. We have 3 leftover questions. We need to collect your thought. For the physical attendees, please hold your patience. We are almost like 1.5 hour away from the end of the game. Thank you very much.